Thanks, Jan, for the very kind introduction and for inviting us here today and for organizing more broadly this series. So, an overview of the RALPH NMR Lyon facility. I'll start and presenting the facility and our expertise, and then Tanguy will present a case study which used to be, which started as an access project within the iNEXT with iNEXT framework and developed into a larger multi-partner collaborative project, which perfectly illustrates, I think, the capacity, the potential of solid-state NMR of our facility in structural biology. Let's start with RALPH NMR. As Anne said, the partner that plays in iNEXT-Discovery for NMR in France is a two-headed partner is the Rhône-Alpes large-scale facilities, Lyon and Grenoble. So the two towns are about 1 hour drive away from each other. And they cover the solution NMR for the IBS and the solid-state NMR access for the Lyon section. The two partners have worked together since 2006 in the context of European projects. So this is the Lyon laboratory. So it's a dedicated building which opened actually its door in 2008. Is 1,500 square meters of spectrometer hall, and about the equivalent space divided into offices and preparation laboratories. It's a very nice and fancy building that typically users and collaborators appreciate a lot. But since last year, I mean, has been much more, sort of away from many visits and collaborations. This is the staff that is hosted in the laboratory, so highly qualified staff, and in the bottom part of the slide, and in the top part of the slide is the plan of the spectrometer hall. So as you can see, a rich collection of NMR spectrometers, 7 NMR spectrometers of different fields, ranging from 400 megahertz to 1,000 megahertz, 1 gigahertz. The gigahertz has been the first gigahertz machine installed worldwide, and was the only one installed till a couple of years ago, when other 1,000 megahertz and the first 1.2 gigahertz have started to be installed in Europe. The personnel working in the center, so it's working on the development of NMR spectroscopy, on the methodology associated with NMR spectroscopy in these different areas of research in chemistry, biology, material science. And the laboratory has hosted 4 ERC grants, 1 advanced, 1 consolidator, and 2 starting grants on the development of the NMR spectroscopy over the last few years. The 2 machines, out of our 7 machines, the 2 machines which are open for access for about 35 access days per year within the iNEXT-Discovery in Lyon are the highest field, the spectrometer, the 1,000 megahertz, and the 800 wide-bore DMP. The 800 wide-bore DMP is a solid-state only oriented machine, while the gigahertz machine is equipped with both probes for solution NMR and solid-state NMR. In the context of our particular expertise within the iNEXT project, I will try to put the accent and explain a little bit the solid-state NMR instrumentation. Actually, I would like to explain why this large collection of accessories, the NMR probes, where we have this list of accessories with different nuclei we can look at, but also different diameters. This is something which is particularly important for the solid-state research. But let's step back and try to give you an idea as why is solid-state NMR needed. So why do we need to differentiate? Why isn't NMR, so solution NMR, enough? Well, because there are a number of systems like viral nucleocapsid, insoluble fibrils, membrane proteins in membranes, in lipid bilayers, which cannot be solubilized easily or cannot be solubilized at all. And for this target, solution NMR, therefore, is not suitable. There are also other classes of molecules, like large protein assemblies or protein of large molecular size. And in this case, the object of interest may be solubilized, but as illustrated here, the quality of the NMR spectra in solution degrades very rapidly with the size of the molecule, therefore of the assemblies. For molecules of weight of 50 kilodalton or larger, as you can see, the one-dimensional spectrum, which is the basis, the root for the acquisition and interpretation of much more complex spectra, degrades to the point that cannot yield any more information, size-specific information, which is the key of the NMR analysis, and cannot be used. So solid-state NMR is the technique which addresses these two barriers: insoluble targets and large proteins or protein assemblies, which cannot be characterized by solution NMR. So it's solid-state NMR has grown over the last 20 years from a small little sister of a younger sister of solution NMR, biomolecular solution NMR, into an adult technique nowadays that can address pertinent answers for these systems. Is it easy to do solid-state NMR? There is a trick. You may have heard about magic angle spinning. What's this story? So in solution, top left here, a small molecule like alanine, for example, gives a very sharp spectrum, 2 lines, because in the molecule there are two families of proton signals, for example, giving this very easy spectrum. The equivalent molecule in the solid, the right top right, in a solid powder is extremely is weird. There's this very broad, large signal, featureless, out of which is very difficult to extract some information. This is because the spins, the nuclear spins inside our magnetic fields, are talking with each other, and they are sending a lot of messages, and these messages are not averaged and simplified by the rotation of a molecule, which in the solid is absent, differently from solution. And the spectrum, therefore, collects a memory, brings a memory on all this interaction, all this noise, which therefore broadens the signal significantly. The traditional way, the established way to recover resolution is to place the sample into a cylinder, the rotor, which is oriented at a particular angle with a magnetic field, and rotates regularly during the NMR experiment. So it rotates within the magnet, within the NMR spectrometer, and as you can see, there is the coil that we use to irradiate the sample and to detect the information, the messages from the sample is surrounding this rotor, where the sample has an homogeneous movement, and you can see the effect of this movement around this magic angle is to turn the static broad spectrum, which is exemplified here by the carbon spectrum of the same glycine here, into at faster span into a series of sharp signals separated by the spinning frequency. And when the spinning speed increases to a certain point, for example, 10 kilohertz, 10,000 rotations per second, so it's quite fast and regular has to be the process. The spectrum sort of is reduced to very sharp signals similar to the equivalent sharp spectrum that one may get from a molecule in solution. Now, the magic angle spinning is it's an old technique. Was invented at the end of the 50s and progressively implemented into what is today modern and NMR spectroscopy. But this as actually the way this averaging is performing actually depends on the kind of nucleus we want to look at. It works, so the frequency like 10 kilohertz are enough to average the lines for carbon nuclei or for nitrogen nuclei, but are not enough to average the lines, the spectra of protons. Protons are the main players in structural biology and are the most interesting nuclei for NMR investigation. So if we look at this figure, this slide here, this illustrates how solution NMR works or has been working now for decades on the left, and how solid state NMR has been working, and that's over the last at the beginning of the biomolecular NMR era of solid state NMR on the right. So on the left, we can see that for a small molecule, this is GB1, so a small size model system. This is you just need about 1 milligram for solution NMR to get what is called the fingerprint, the solution HSQC, the proton-nitrogen correlation. So it's a collection of signals where every residue, every amino acid is represented by one peak, one signal in this map. So this is the starting point for assignment and the structural calculation, structural determination, and for all the study of a molecule in solution. This is extremely rapid to acquire. This is the basic spectrum that you typically get in 10 minutes. And that's the key of the success of solution MR for the analysis of a molecule in solution. The fact that the available spin rates up to last few years were only 10 or 20 kilohertz limits the capacity of averaging of the resonances in solid state NMR. And on the right, you can see the equivalent correlation for a molecule, which is the same molecule crystallized in microcrystalline form. So as you can see here, the signals are not resolved; they are highly overlapped, and the spectrum is not usable. So a key in the technique has been brought by instrumentation. So is it possible to spin faster? Well, to spin faster, and this is a little bit the inside of what the instrumentation is. So you have this rotor which has its cap has a propeller shape. You can see it here on the top, and it's inserted so inside what we call a stator, which is a device which has some gas inputs, so the bearing gas, the drive gas, and the variable temperature gas. So with this pneumatic system, you are able to lift the rotor. And this is what one of these gas flows does, the bearing. And and at the same time, another flow is produced tangential, and it's the cap, the tubing cap, and produces the rotation. So it's a purely pneumatic rotation. So the rotor levitates and rotates. So it's an extremely sophisticated technique. And during this rotation, stability can only be achieved if the surface speed of a turbine blade tip is smaller than the speed of sound. So if we approach speed of sound, we have some shock waves which destabilize the rotation and results in a rotor crash. So all this works, provided therefore that the linear speed on the top of the rotor, at the edge of the rotor, is smaller than the speed of sound. Which means that is it possible to spin faster? Well, it is possible, provided we reduce the diameter of the rotor. Therefore, reducing the diameter of the rotor is further efforts in miniaturization, but also in reducing the amount of sample that we can look at. And for many years, this has been so the technical problems, but also the conceptual problem of reducing the sample volume and therefore reducing sensitivity has been an obstacle for the development of faster rotation on smaller quantities. So this has made a major step forward in the last 10 years with the development, first of a so-called 1.3 millimeter rotoscopable spinning at 60 kilohertz, then more recently with a 0.7 millimeter rotoscopable spinning almost twice as fast, between 100 and 110 kilohertz. So with this rotors, you can see that here the decrease in size. It is possible to much better average this magnetic noise between magnetic signals, magnetic crosstalks between nuclear spins inside the sample, and come from an unresolved proton spectrum on the left here, on the bottom, to a resolved proton spectrum with several lines for a protein here on the right. So this is a spectrum of our work, work in our facility in 2016, 2015, 2016, when we received the first prototype of the Bruker probe. Since then, the field is evolving. Today, there are examples of probes spinning under 50 or under 60 kilohertz in Zurich with Ago Samuelsson and Beat Meyer. The result is here that in a sort of magical way, we can also in the solid, in a microcrystalline sample, transform the very broad unresolved spectrum on the left to a very resolved solution-like spectrum, where again we can track the position of all the signals from all the residues in the molecule. So there is what we call the nice resolution. So it's still tens of Earths, but all the signals, most of the signals are baseline separated. And what is most importantly, we can produce this reduction in volume without affecting sensitivity. And the acquisition of solid-state NMR spectra, which is typically for many years was had to rely on the acquisition of heteronuclei, insensitive heteronuclei like carbon or nitrogen, is today possible with this developments. It's today possible on protons, so the most sensitive and abundant NMR nuclei in a sample. And a spectrum like this, also in the solids today at high field, can be recorded in 10 minutes on a limited amount of samples, half of a milligram. So we are really comparable to the solution NMR requirements in terms of samples. In this process, the field have the high magnetic fields have an importance, and you can see here the fact that resolution improves going from the 500 to 1000 megahertz. You can appreciate it on this spectrum of the side chain. This is proton-carbon fingerprints of a side chain of a model GB1 molecule, but you can also more importantly, more highly appreciate it on the spectrum of a large bacterial transporter in lipid bilayers, as the one is exemplified here. You can see a spectrum which is completely unresolved on the left, and several signals, therefore a lot of site resolution, a lot of potential for site-specific investigation on the spectrum on the right. And you can also appreciate the difference in sensitivity, and therefore the different experimental time from several hours to only 2 hours in this highly diluted sample, like the bacterial transport. So once you can achieve this spectra, the workflow of solid-state NMR is very much similar to what has traditionally been developed for solution NMR. So out of these signals, we can monitor specific pairwise proximities between pairs of nuclei. So starting from a random starting model, we can use this information to restrain the the protein folding, simulating annealing protocols, and in an iterative cyclic manner, we can calculate structure. So with this new methodological developments, we can today apply exactly the protocols and the workflow developed for solution NMR. And an example here is summarized in this slide. And this is typically what we propose to our users when we come with a new samples. We need to acquire backbone resonance assignments, so give a name to all the resonances. There are a number of techniques which can be acquired efficiently for performing this action in the solid state. We can give a name to all the resonances, also for the side chains, not only for the protein backbone, but also for the different side chains with other particular spectra. And then we typically record a number of multidimensional correlations, 3D or even 4D correlations, which are able to, where every cross signal, every peak, every peak in this spectra represent pairwise proximities, which are then interpreted nowadays almost automatical. In an automatical way, we have routines that were developed, like Sayana, by Peter Gunther, developed for solution NMR, which can be used almost in an identical way today for calculate structures in the solids. Now, what's also interesting is that also in the solid state, NMR keeps its unique potential to integrate structural information with information on dynamics. Dynamics happening over different time scale, from very fast Meteor rotation in nanosecond to picosecond range, to loop motions, side chain rotations, or local unfolding. So dynamics of a very vast window timescale from picoseconds to seconds. And the solid state NMR, in analogous ways to what solution NMR, can do that by reading, associating to each of the signals in an NMR spectrum. Here, for example, the blue and the red signal. For each of these signals, NMR is possible to read the, I'm using technical terms, the anisotropy of the particular property, some particular properties of the nuclear spin, of the nuclear shifts, of the nuclear Hamiltonian, and evaluating the way they are modified by local motions or scaled down by local motion. There are a number of experiments that are sensitive, therefore, to motions, in this case, happening in a particular time scale, or evaluate the capacity of local motion to produce, to enhance nuclear relaxation, which is another phenomenon that we can measure site-specifically in solution NMR, but also today in solid-state NMR, and out of which we can get selective information on fast or slower timescale. Now, the interesting of solid-state NMR is that, therefore, today, this adult technique can perform as nice as solution NMR, but we can finally tackle all the substrates which were precluded in the traditional solid-state NMR analysis. We can study samples which are solid or crystalline. Crystalline, why to study samples which are crystalline? The crystallographers do that much better. Yes, sure. Crystallographers can do it better if you can get a large single crystal. The nice thing of NMR is that NMR is not sensitive to global order, but only to local order. So provided you get a crystalline powder, a nanocrystal, so which is still diffracting, so it's a diffracting powder, the quality of the spectrum, this is the case for GB1, what is a milestone achievement of Rachel Martin and Kurt Zilm back in 2003, the quality of the spectrum is identical for this for different preparations, which means that the solid NMR can integrate, therefore, crystallography already on a number of studies, samples for which you cannot obtain large single crystals. So a typical workflow in this case is to precipitate get the molecule in solution, precipitate with PEG or MPD, centrifuge directly the precipitate into the NMR rotor, and produce NMR. And already here, there are a number of surprises. This is, there are two papers. Actually, this is just 2019, one on the left, and just 2020 on the right, where two well-known enzymes, human carbonic anhydrase and human copper superoxide dismutase. Actually, the community, so is Rasmus Linse on the left and us on the right, the community has realized actually that very well-known molecules for which a lot of crystal structure have been determined, solved. There are no surprises. The molecules were thought to be extremely rigid. Actually, at the active site, inside hydrated crystals, these molecules contain a lot of dynamics, and this dynamics is relevant for function and can be associated to particular catalytic acts. And both on the left for human carbonic anhydrases next to the catalytic zinc ion, and the same equivalent phenomena we found on the copper zinc superoxide dismutase, highlighting therefore differences between molecule with and without copper, without on the top, with on the bottom. There is dynamics, and this dynamics has a role in catalysis. This is all in the folding, the stability of the molecule. This is typically overlooked by simple crystallographic studies. But apart from that, solid-state NMR can work on non-crystalline samples. For example, fibrils. This is an example of so amyloid fibrils is an example from a user of a platform where, again, proton-detected solid-state NMR was used to transform this kind of this STEM image into an high-resolution structure of the assembly. We mentioned the size, the problem connected to the size of the molecule, and here there are a number of recent examples where large molecules, these large globular proteins on the left, but also assemblies, non-crystalline assemblies like measles nucleocapsids, HIV tubular assemblies, protein RNA complexes, can be immobilized either by precipitation or simply by centrifugation due to their large size. This was one of Ivano Bertini's intuitions back in 2011, and can be immobilized and become suitable for solid-state NMR investigation. And finally, the class which is today maybe the most exciting application area of solid-state NMR is membrane proteins in lipid bilayers. So no need to solubilize the molecule in detergents, but solubilize the molecule but may alter its structure and its properties. Solid-state NMR can directly reconstitute these molecules in lipid bilayers, and which can be dialyzed. So lipids can be dialyzed in, exchanging detergent into lipids. They yield a pellet which can be centrifuged into the NMR rotor. And here there are three examples of three membrane proteins for which we have contributed to the structural determination in our facility. And I'll now pass the
Okay. Thanks, Guido.
Stage to Tanguy.
Yes. So now I will present a project which started as an access to our platform by Tom Schwarzer and Catherine Castiglione, and as continues as iNEXT, so access from Lauren Andreas, and was a collaboration between, at the end, finished as a collaboration between the three sites. And why we are talking about that is because it will give a quick overview of the possibilities raised by modern mass NMR for tackling structure, dynamics, and interactions for proteins. And this project was focused on the outer membrane protein alkyl of Pseudomonas putida, which allows to the small hydrophobic compounds such as well, alkanes or, for example, here carbon, to enter the cell. And the mechanism of this transport was unknown before that. So yes, this moves. Okay, good. So all the sample preparation was done well. Was done also in the we provide some access to the biolab also to our user, and we can do sample preparation for them by over expressing the protein, for example, here in E. coli, doing purification detergent and for membrane proteins, and doing reconstitution into lipids, and then centrifugation into the mass NMR rotor, which are some specific step for mass NMR. And also one important thing is to optimize the sample. For this, we can record, we can use NMR to optimize the sample by recording 2D correlations of the protein, here correlating nitrogen and proton, for different types of lipids, until we find good lipid conditions that yield result in sensitive spectra. And then we can optimize the protein to lipid ratio, so to have an optimal sensitivity of the NMR experiments while keeping the protein intact, as observed here, for example, by the signature spectra that are absolutely identical between the two different concentrations. So this is the same spectrum acquired with a higher magnetic field and faster MIS, where we see that many peaks can be isolated. So we performed, we determined the structure of alkane both in micellar solution and in lipid bilayers. And actually the transmembrane part of the protein is preserved between the two environments, but something that shows the importance of the membrane mimicking environment is that the extracellular loops of alkyl in solution are actually disordered, while in solid state they elongate the beta barrel. And this is actually important because it forms an extension of the pore where with reverse polarity, where the hydrophobic residues are pointing inwards, while the hydrophilics are pointing outward. And this allows hydrophobic substrates to come inside the pore. However, in the structure, a striking feature is that, the striking feature of this structure is that the pore is actually closed in the middle of the transmembrane. And this was it could be quite puzzling, but we were trying to see if something else can be determined on this protein more than the static structure. And for this, we measured spin relaxation parameters that are sensitive to microsecond motions, and show that at the interface between the beta transmembrane barrel and the extracellular extension, there is some interface part where there is actually quite a lot of dynamics, and this raises the question if the substrate cannot exit the protein to the membrane part, hydrophobic part of the membrane, by a dynamical lateral exit. And this was further checked by probing interaction with of the alkyl with a substrate, which is here carbon, and by mapping the chemical shift perturbation in presence or in absence of carbon on the structure, and we see a nice correlation. Well, we see also interaction with the substrate at this interface between the transmembrane part and the extracellular one. This was further demonstrated by molecular dynamics simulation performed by collaborators in MPI. that showed that there is a dynamic remodeling of the pore in the presence of the substrate. That here in the video that shows that the pore can widen to allow the substrate to go out. So finally, this was the mechanism of the protein was unknown. How can hydrophobic molecules can pass through the lipopolysaccharide layer? And here we showed that by mass NMR and molecular dynamic simulation, we showed that the structure of alkyl can allow the passage of these hydrophobic compounds, clustering in the extracellular loops of alkyl first, and then being released little by little into the membrane bilayer. So with that, we would like to thank people from Guido's group, in particular Lauren Andreas, Tobias Schubis, and Jan Stanek that worked on the alkyl project, and all the people from Lyon, from Göttingen, Florence, Munich, Hyderabad, and also people from Bruker Biospin.
And of course, for iNext and iNEXT-Discovery, and please submit a project. I hope we are convinced Solista NMR can do something for you.